All right, today we're going to go over the answers for case study number 12. So first you were asked to define some terms. It started with MHC class 1. So this is a molecule that presents internal peptides to CD8 T cells, and it's expressed on the surface of all cells, or, pretty much all cells. There's just a couple of exceptions, like RBCs. Now, MHC class 2 is a molecule that's expressed on the surface of antigen-presenting cells. So it presents external peptides. So these are peptides from, pathogens that are gobbled up by APCs. They break them down, and then they present these peptides on MHC class 2 to CD4 T cells. And then, so a CD8 T cell is a killer T cell or a cytotoxic T cell. It destroys other cells with enzymes. It recognizes MHC class 1 on other cells. And then, of course, CD4 is a T helper cell. So this is the T cell that binds to MHC class 2. And so T helper cells are involved in activating other cells, so that they fight an infection, especially when it comes to B cells, activating B cells to make antibodies. And then TAP1 and TAP2, so these are genes that are talked about in this case study. So the molecules that the gene codes for, they are transporters of peptides that are going to be loaded on MHC class 1 type molecules. And in order for an MHC class 1 molecule to be expressed on the outside of a cell, it has to first be loaded with a peptide. All right, what is the technique used to examine how much MHC1 is present on T cells? That would be flow cytometry. And what are two cytokines produced by CD8 T cells that activate inflammation? That would be interferon gamma and TNF alpha. All right, question 3. Why do Tatiana and Alexander have low numbers for CD8 T cells but high levels of CD4? Excuse me. So the idea here is that early on in development, these T cells will express both CD4 and CD8, and it's during the time that they are maturing in the thymus that they become either dedicated CD4 type cells or CD8 type T cells. And it has to do with which one of those MHC classes they bind to in the thymus when they're being educated. So any given uneducated T cell, like I said, it expresses both CD4 and CD8. It has the potential to be either a CD8 T cell or a CD4 T cell. But depending on, so there's dendritic cells and epithelial cells in the thymus that these T cells bind to. So if it binds to a MHC1 molecule in the thymus, it becomes a CD8 T cell. If it binds to MHC class 2, then it becomes a CD4 T cell. And so when the fact that Tatiana and Alexander have low numbers of CD8 T cells but high levels of CD4 T cells, They have no issue with producing these uneducated T cells. What they have an issue with is the expression of MHC1. It's really low. So these T cells that are formed, they're going to there's more MHC class 2 to bind than MHC class 1 in the thymus. So we end up with high levels of CD4 T cells and really low levels of CD8 T cells. So number 4 asks, why are Tatiana and Alexander the only children in the family affected? And what does this have to do with figure 12.7? Figure 12.7 is showing us, I believe that's the one that is that's the one that's showing the inheritance of MHC haplotypes in this family. And so Tatiana and Alexander both have 2 copies of the same haplotype. And so, this would help. So originally in the case study, it was talking about the fact that this MHC haplotype, there might be an issue with MHC molecules themselves, the genes for that produce those proteins. And it turns out those are fine, but also in this same genomic region, in the same, in fact, that's what we would see in figure 12.3. That's also where the TAP genes are found. So the fact that they both have 2 of the same copy of this haplotype that also includes the same copies of these TAP genes, it turns out that the TAP2 gene copies that they have a nonsense mutation. And the other 3 children are not affected, and the parents are not affected because nobody else has 2 copies of that defective TAP2 gene. So why does a deficiency in TAP1 or 2 cause no MHC class 1 molecules to be presented on the surface of cells? It's kind of like what I was talking about earlier. If there's no peptide loading onto MHC1 molecules, then those molecules are not expressed on the cell surface. So with this, with these deficiencies in these transporter proteins, TAP1 and TAP2, that form a heterodimer in the ER, and MHC class 1 is loaded by, or is loaded in the ER lumen with these peptides. So since there's this mutation in TAP2, then MHC class 1 does not receive these peptides, and so it does not get expressed on the outside of the cell. And why do Tatiana and Alexander have problems clearing encapsulated bacteria? What that has to do with is the fact that they've had these repeated infections that have actually caused damage to their airways. They have a condition where the airways are they're wider than they should be. They are going to be thicker, and mucus, which usually mucus can be readily expelled from the lungs, and mucus traps encapsulated bacteria, and that's one of the ways that encapsulated bacteria, that's how it's cleared from the airways. But what can end up happening is mucus ends up pooling in the lungs and along with these encapsulated bacteria, so they're just kind of like they're constantly being exposed to these bacteria, so they're just constantly having infections by these encapsulated bacteria due to the damage from them having these viral infections. Why do you think Tatiana Alexander seemed to be able to fight off some types of viral infections but not others? That's not completely understood. There are a few different ways that they might be able to do that. In the case study, it mentioned the idea that maybe certain types of antigenic peptides might trigger a greater expression of MHC1 than others, which would mean that they could mount, responses to some viruses better than others. Or it could be that extracellular virus is being taken up by phagocytes. B cells are being stimulated, and so then there's neutralizing antibodies being produced, which could fight certain viruses, and also maybe it could be natural killer cells, which are also involved in fighting viral infection. And that's it.